In this video, we are going to see about previous year question concept series from the topic of regeneration. And you are watching this on Life Science Library. First question from Hydra. In this case, major head induced of hypsome organizer in a set of WNT protein acting through canonical beta-catenin pathway. What could be the result if transgenic hydra is made to globally misexpress downstream WNT effector beta-catenin? So, consider it as an hydra. It consists of an hypsome organizer with some tentacles. For this head portion anterior body to be expressed, generally it needs WNT signaling beta catenin. This is the concept. They are giving us a condition where what could be the result if transgenic hydra is made to globally misexpress the downstream WNT effector of beta catenin. So here we are speaking about canonical beta catenin pathway. If considering a case wherein if downstream effector molecule is globally misexpressed, it is highly expressed or inhibited, globally misexpression, not inhibition, globally misexpressed, very high expression or abnormally express of WNT beta catenin, what will be the result? The result will be excess growth, that is ectopic buds will be formed all along the body axis, even on the top of newly formed buds, because excess, that is misexpression or abnormality in expression is seen. So A is the right answer. Next question. Given below are some of the statements regarding regeneration. A type of regeneration characteristic of mammalian liver is considered as compensatory regeneration because the cell undergo redivision and they enter into cell cycle to compensate for the cells that are lost in mammalian liver. A is the right answer. This is the concept of compensatory regeneration. Regrowth of hair shaft from the follicular cells exemplifies the stem cell mediated regeneration. Correct. Regeneration occurring through repatterning of existing tissues with little or no growth is called morphollaxis, and this kind of regeneration is seen in hydra. C is the right answer. Adult structures undergoing dedifferentiation forming blastema and then redifferentiate to form a lost structure is called as epimorphosis. So epimorphosis is a condition where a cell is dedifferentiated and then they are redifferentiated to form desired tissues and cells. This kind of regeneration is called as epimorphosis. So all the four statements are correct. D is the right answer. Next, June 2018. Which of the following statements regarding limb regeneration is seen in salamander? First statement, a normal limb is regenerated after amputation irrespective of whether the cut was made below the elbow or through the humerus. Second statement, it occurs by compensatory regeneration. No, compensatory regeneration occurs in mammalian liver. Regeneration occurs through the formation of blastema. It is essentially consists of an unspecified multipotential progenitor cells. Fourth statement, proliferation of blastema cells does not require nerves and factors secreted by nerves. No. D is not right answer. Regeneration is occurred after amputation irrespective of whether the cut was made below or above the elbow. This is right. Do blastema formation occur in salamander? Blastema formation does not occur even in mammalian liver. We have studied that mammalian liver consists of compensatory regeneration where blastema formation does not occur in mammalian liver. A is the right answer. Salamander is associated with scar-free wound healing. Compensatory regeneration of liver is also associated with scar-free. That is, scar will occur in liver in case of fatty liver or other conditions where the liver is damaged. So maybe the compensatory regeneration, blastema formation does not occur, and the salamander undergoes the scar-free wound healing mechanism. So it is not that much associated with blastema. The correct answer is A. What could be the effect on the new limb regeneration if more than 90% of nerve supply is severed before amputation? So apical ectodermal cap stimulates the growth of blastema by secreting FGF8. But regeneration does not take place. A is the right answer. Now comes to a question from Dictyroxylum discoideum. In Dictyroxylum discoideum is an social amoeba. We will study whether how it aggregate, signaling mechanism mediated by cyclic AMP, how it handle an unfavorable condition, how it adhere during aggregation. So we will study all these are the major parts of how Dictyroxylum discoideum has to be studied. Now coming to this question. We will very well say that Reg A is an extracellular phosphodiesterase. Correct? Every amoeba at the time of aggregation has the potential to make and receive and rely on cyclic AMP. A is right. So this is very static, that doesn't need much understanding. You should know that it is associated with cyclic AMP. It involves in aggregation and migration during unfavorable condition, and it is social amoeba. It is an asexual condition. It forms spores and pre-stalks. It's called a slime mold. These are the basic detail and how the signaling occurs. It is a basic cyclic AMP signaling pathway where the CAR1 is involved in activation of this pathway. This is the basic understanding. A and D is the right answer. A is the right answer. June 2018 Dictyroxylum discoideum question. June 2013. Hydra shows morpholactic regeneration and involves which of the following signal transduction pathway in its axis formation? Now we have seen in morpholactic regeneration in Hydra. It forms WNT beta catenin signaling pathway. So A is the right answer. Next, from morpholactic regeneration of Hydra. Morpholactic regeneration means repatterning of existing tissues with little and new growth. This is the basic definition of morpholactic regeneration. So there is nothing much great to understand better. This is the answer. E is the right answer. Next, December 2009. Again, morphollaxis can be defined as reinitiation of cell division in existing cells followed by repatterning. Just now we are studied. B is the right answer. So far we have seen, in my knowledge questions, all the questions from CSIR net from 2020 to 2008 in my knowledge. These are the questions from the question bank issued from previous year questions from regeneration. Basic thing of regeneration is you should study about Hydra. We have seen questions from all these. Salamander and liver. It is compensatory regeneration. There are types like morphollaxis. You study about morphollaxis, epimorphosis, then compensatory regeneration, then stem cell mediated regeneration, and case based studies. We have seen in Hydra where if WNT is not present, what will happen? If they are transplanted, head portion is being transplanted in the foot plate, what will happen? If the nerve cell is cut, is amputated, whether the regeneration will occur or not. These are the cases by which one candidate should study regeneration. Hope so this topic is very clear from developmental biology. Unit 5 Csirnet Life Science portion. If you think this video is very useful, please do like, share and subscribe to my channel and refer this video with your friends and fellow aspirants. Thank you.